The overall aim of the following experiment is to explore the electronic structure of isolated gas phase molecules and clusters with molecular beams and synchrotron radiation. This is achieved by generating a supersonic expansion of a molecule seated in a carrier gas in a molecular beam apparatus. As a second step, the resulting molecular beam is integrated with synchrotron radiation and mass spectrometry is performed. Next, a photoionization efficiency curve is generated in order to decipher and quantify the electronic properties of the studied system. Results are obtained that can be compared to theoretical calculations, and subsequently the electronic structure of molecules can be determined.
The main advantage of this technique over others is that synchrotron radiation allows a very selective yet universal ionization method to probe molecules and clusters in the gas phase. We hit upon this idea a few years ago since we wanted to get DNA molecules into the gas phase and measure the ionization energies. This method provides unprecedented access to electronic structures of molecules and clusters, particularly when one does photoionization of DNA bases.
To begin, remove the back flange and disassemble the 3/8 of an inch stainless steel tube from the apparatus. Make sure it is clean and the 100 micron orifice is clear by looking through it at a light source. For cleaning, fill the tube with approximately 1 milliliter of ethanol and scrub the inside using cotton tips. Then, dry the tube with compressed air. To avoid blocking the orifice,